Mendelian disorders, example hemophilia, color blindness, sickle cell anemia, thalassemia and phenylketonuria. Then chromosomal disorders, Down syndrome, Turner syndrome and Klinefelter's syndrome. The change in the chromosome number happens in an autosome. So it is called as autosomal disorder. What is the phenotypic ratio of dihybrid cross? 9:3:3:1. And what is the genotypic ratio of the dihybrid cross? 1:2:1, 2:4:2, 1:2:1. Hi, hello everyone. Welcome back to yet another revision class of Chapter 4 of 2nd PUC Biology, Principles of Inheritance and Variation. Myself, B. Shobarani from the Department of Biology, Vidhyaashram Pre-University College, the Temple of Excellence. This particular chapter, Chapter 4 of 2nd PUC Biology, called Principles of Inheritance and Variation, has a weightage of 14 marks. So it's a very big marks. So the concepts what I explained in this particular chapter is very, very important from examination point of view. So what am I going to discuss in this particular session? I'm going to discuss about the questions that were repeatedly asked in the previous question papers pertaining to this particular chapter called Principles of Inheritance and Variation. Moving on to the flow chart of this particular chapter, Mendel's law. As we know, genetics is what? Genetics is nothing but the study of heredity and variation. Heredity means what? Inheritance of characters from one generation to another generation. So we all know pretty sure John Gregor Mendel, who is called the father of genetics, came across all his ideas were put across in the form of laws called Mendelian laws of heredity. So law of dominance, law of independent assortment and law of segregation. Moving on to the exception of Mendel's law. Other than there was a slight deviation from Mendelian laws, that is incomplete dominance, which was studied with a plant example called snapdragon, codominance that is particularly AB blood group, and multiple allelism, that is when genes occur in more than one form. The classical example is ABO, occurrence of ABO blood groups, then pleiotropy, then polygenic inheritance. Then mechanism of sex determination can be studied in two ways. One is called male heterogamety and the other one is called female heterogamety. So what are the examples we are going to study in male heterogamety, XXXY type, taking the example of Drosophila melanogaster, which is called the Cinderella of genetics. Then XXXO in grasshopper. Female heterogamety, ZZZW type, ZZZO type. So this is very important from examination point of view. Definitely one question will appear regarding this concept, that is mechanism of sex determination, either from male heterogamety or female heterogamety. Moving on to the genetic disorders. So the genetic disorders were classified into two disorders called the Mendelian disorders and the chromosomal disorders. What were the examples that were quoted for Mendelian disorders? Hemophilia, color blindness, sickle cell anemia, thalassemia and phenylketonuria. Then chromosomal disorders, the disorders that happen due to change in the chromosomal number. Down syndrome, Turner syndrome and Klinefelter's syndrome. Down syndrome is also called as 21st trisomy. And Down syndrome is common for both male and female, whereas Turner syndrome is the syndrome which occurs only in female. And Klinefelter syndrome is a syndrome which occurs only in male. So these are the different types of genetic disorders. That is Mendelian disorders, example hemophilia, color blindness, sickle cell anemia, thalassemia and phenylketonuria. Then chromosomal disorders, Down syndrome, Turner syndrome and Klinefelter's syndrome. Then moving on to the MCQs. Just have quoted one MCQ question, that is a multiple choice question, like what type of MCQs might appear from this particular chapter, which is having a weightage of 14 marks. Who noted that the behavior of chromosomes was parallel to the behavior of genes? Was it T.H. Morgan or Gregor J. Mendel, Alfred Stutwald, Walter Sutton and Theodore Boveri? So the option is D. Walter Sutton and Theodore Boveri. So like this you might get MCQs from this particular chapter called Principles of Inheritance and Variation, which is having lot of weightage of marks of 14 marks. Then moving on to questions like point mutation arises due to the change in single base pair of DNA. Mention the 4 symptoms of Down syndrome. Already I told you Down syndrome is a chromosomal syndrome which may occur both in male and female. It is common to both male and female, which is also called as 21st trisomy, where the chromosomal complement will be 45 plus XX is equal to 47 or 45 plus XY is equal to 47. So this Down syndrome is an autosomal syndrome. The change in the chromosome number happens in an autosome. So it is called as autosomal disorder. So what are the symptoms that appear in a person who is having Down syndrome? Short stature with small round head, furrowed tongue and partially open mouth, palm is broad with characteristic palm crease, then physical, psychomotor and mental development is retarded. So they are mentally retarded. So the symptoms of Down syndrome and the chromosomal complement is 45 plus XX is equal to 47 in case of females and 45 plus XY is equal to 47 in case of males. As I said, this is also called as 21st trisomy because trisomy means what? 2n plus 1. And the change that plus 1 happens to the 21st autosome. Hence, it is called as 21st trisomy. So, the symptoms are, all these are the symptoms. Moving on to question number 4. Explain the sex determination in honeybees. We already studied about male heterogamety and female heterogamety. So, sex determination in honeybee is based on the number of sets of chromosomes, whether it is haploid or diploid the individual receives. So, here the female is diploid. Diploid means how many chromosomes? 32 chromosomes. Whereas male is haploid, having half the number of chromosomes, that is 16 chromosomes. An offspring formed from the fertilization of a sperm and an egg developed into either queen or worker. Both are females. Anyhow, in the coming slide, I have given the schematic representation of sex determination in honeybees. So here, an unfertilized egg develops into male, that is drawn by means of parthenogenesis, direct development. So without, an unfertilized egg develops into male. This method of sex determination, that is the sex determination in honeybee, is called haplodiploid sex determination because it involves what? Both haploid set and diploid set. Male produces sperms by mitosis and female produces egg by meiosis. Hence, the male honeybee don't have father and thus cannot have sons but have grandson. So, how do you represent this sex determination in honeybee? This is the representation. The parents female 32, that is I said diploid set, and male haploid. Females undergo meiosis, so 16, 16, so divide into, and there it is mitosis. So here you can see the male, only 16, half the set, haploid set of chromosome will be formed male, and 16 crossed with another 16, it is 32, that is female. Here it is male and here it is female. This is a very important question from examination point of view. Definitely this is the one which commonly appears in the examination. Moving on to the next question, very important question from examination point of view. So, state the law of independent assortment. Write schematic representation of inheritance of two genes. So, whenever they ask inheritance of two genes, obviously it is a dihybrid cross. So, what is a dihybrid cross? It is a cross between two plants differing in two contrasting characters. One gene inheritance or monohybrid cross, it is a cross between two plants differing in only one contrasting character. So, what is the statement? And this dihybrid cross is verified by Mendel's law of independent assortment. So, what does the law state? When the alleles for more than two contrasting characters are brought together in F1 hybrids, they assort independently by one another in F2 generation during the formation of gametes. So how do you represent this? So this is the schematic representation. So this is the schematic representation. So parents, as I said, dihybrid cross is a cross between two plants differing in two contrasting characters. So one character is round, opposite for round is wrinkle. Then one with, this is shape and this is color. So with respect to shape, round, opposite for that, wrinkle, color, yellow, dominant is yellow, and for that it is green. So how do you write the genotypes? Capital R, capital R, capital Y, small r, small y, small y. So when you cross this, so these are the gametes. So you can write it as capital R, capital Y, small r, small y. So go for the F1 generation, first filial generation. So you get capital R, capital Y, small y. So here all F1 offsprings are round yellow. So all are round yellow. So F1 offsprings are round yellow. How do you go for F2? F2 is obtained by selfing F1. That is F1 into F1. So this one into again one more F1. You get F2. When two F1 individuals are self-crossed, that is capital R, capital Y, small y, that is heterozygous round yellow, crossed with capital R, capital Y, small y. So we'll move on to the next slide. So here this is the Punnett square. So how do you write the checkerboard or the Punnett square? So this is how you are going to write. So you take gametes, male gametes on one side and female. So this is one F1 and this is another F1. So when you take, what are the gametes? Capital R, capital Y, that is round yellow. Then round green, capital R, small y means that is round. This is wrinkled yellow, and this is what? Wrinkled green. So when you cross these gametes, so round yellow means how much? 9 you will get. Then round green 3, wrinkle yellow 3, wrinkle green 1. So here you should understand the ratios, very, very important. The phenotypic ratio is 9:3:3:1. So what is 9? Round yellow. What is 3? Round green. What is 3? That is wrinkle yellow. And what is 1? Wrinkle green. So what is the genotypic ratio? Genotypic ratio is 1:2:1, 2:4:2, 1:2:1. I know it is a little bit difficult for the students to remember. So the best way to remember the genotypic ratio of dihybrid crosses is what? So right hand side 1:2:1 and the left hand side 1:2:1. Middle you need to add 2:4:2. So here 1:2:1, 1:2:1. So middle what you are supposed to add? 2:4:2. So it is easy for you all to remember. So what is the phenotypic ratio of dihybrid cross? 9:3:3:1. And what is the genotypic ratio of the dihybrid cross? 1:2:1, 2:4:2, 1:2:1. Hope you have understood the dihybrid cross, which is very important from examination point of view. Moving on to the next question. T.H. Morgan selected fruit fly for his genetical experiment. Why? Give any 3 reasons. And it is a split question. B. What are chromosomal disorders? Give an example. So why did Morgan select Drosophila melanogaster or the fruit fly for his experiment? Just like how John Gregor Mendel selected the garden pea for his experiment. So what were you have to give 3 reasons. T.H. Morgan worked with a tiny fruit fly which is commonly called as Drosophila melanogaster or the Cinderella of genetics, which were found very suitable for such studies because of the following reasons. So what were the reasons which made Morgan to select particularly the fruit fly for his experiment? They could be grown on simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks. A single mating could produce large number of progeny flies. There was a clear differentiation of the sexes. The male and the female flies are easily distinguishable. So these are the important reasons why Morgan selected the fruit fly or the Drosophila melanogaster for his experiment. So what is the next split question? What are chromosomal disorders? Already we have studied in the flow chart, genetic disorders have been broadly classified into Mendelian disorders and chromosomal disorders. The very word itself will tell you. Chromosomal disorders means what? Change in the chromosomal number. The disorders that are caused due to change in the chromosomal number. The change in the chromosomal number might be to an autosome or to an allosome. So the disorders in human beings are caused due to absence or excess of abnormal arrangement of one or more chromosomes are called chromosomal disorder. So here chromosomal disorders might be due to monosomy, that is 2n minus 1, or due to trisomy, that is 2n plus 1, tetrasomy, that is 2n plus 2, nullisomy, that is 2n minus 2. So here change in the chromosome, there might be an addition or a deletion of a chromosome from the normal set of chromosome which is going to cause the chromosomal disorders. Like what? What are the examples you are going to give for occurrence of chromosomal disorders? Very important is Down syndrome. As I already said, Down syndrome is also called as 21st trisomy, which is an autosomal disorder. And one more, that is Turner syndrome. That is, Turner syndrome is what? It is allosomal disorder. Allosomal. Klinefelter is also again an allosomal disorder. So, what are the examples you are going to give for chromosomal disorder? Down syndrome. Either you can give Down syndrome, Turner syndrome, or Klinefelter syndrome. Moving on to the next question. What are the two types of disorders of human where the karyotype is 47? So already I told you in the previous slide, the karyotype or the chromosomal complement for Klinefelter's syndrome, and the other one is Down syndrome. So 47, the chromosomal, whichever, so the two types of disorders of humans where the karyotype is 47. 47 means what? 44A plus XX is equal to 47. 45A plus XY is equal to 47. So this is Down syndrome. Klinefelter syndrome, 44A plus XXY is equal to 47. So the normal set of chromosomal number, the nucleus of a cell of a normal person will be having what? 46, that is 2n, that is 46 is the chromosomal number, but there is a change here. So there is an addition. This is due to what? Trisomy, that is 2n plus 1. So what are the examples you can give? Klinefelter syndrome and Down syndrome. Moving on to next question. What is phenylketonuria? Write the cause and symptoms of phenylketonuria. So what is phenylketonuria? It is an autosomal disorder. Why? Due to the presence of a recessive gene, and it is an inborn error of metabolism also. It is, you can also call it as an inborn error of metabolism and inherited through the autosomal recessive trait. So that is the reason why we call it as an autosomal recessive trait. The affected individual. So what are the symptoms of phenylketonuria? They are usually called as PKU idiots because they show idiotic behavior, because they have mental imbalance. Hence, we call them as PKU idiots. So, the affected individual lacks an enzyme phenylalanine hydroxylase that converts the essential amino acid called phenylalanine, a substance found in our food, into tyrosine. So, because of the lack of that enzyme, as a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. So this is the cause and symptoms of phenylketonuria. Symptoms. This is the cause. Now what are the symptoms? So what are the symptoms a phenylketonuria person exhibits? Accumulation of these in brain cells results in mental retardedness. Hence we call them as PKU idiots. Phenylpyruvic acid and other derivatives are also excreted through urine because of its poor absorption by the kidney. So it may affect the kidney also. Infants have lighter skin, hair and eyes than their siblings without this disease. So these are all the symptoms. Moving on to next question. Define point mutation. Give an example of point mutation. Change in single base pair of DNA is known as point mutation. And what is the classical example for the occurrence of point mutation? You can give the best example is what? Sickle cell anemia. Then moving on to question number 10. What are the conclusions drawn by T.H. Morgan from the crossing experiments in Drosophila with respect to linkage? So, what was the final conclusions drawn by T.H. Morgan regarding linkage? So, what was his opinion? The higher the linkage, lesser is the recombination, and lesser the linkage, higher is the recombination. In other words, how do you put across this? The genes which are tightly linked will show lesser recombination, and the genes which are loosely linked will show higher recombination. Moving on to question number 11. What is incomplete dominance? Explain it with reference to flower color in Snapdragon. This is also a very important question from examination point of view. As I said that in the previous slide, I said Mendel put all his ideas Mendel's ideas were put across in the form of laws called Mendelian laws, and one law was called as law of dominance. According to the law of dominance, the character which is dominant will express itself in the F1 first generation, and all the offsprings will have the dominant character. But there is a slight deviation from the Mendelian law where the dominant character fails to express itself completely in the F1 generation. Instead, an intermediate character appears, and hence it is called incomplete dominance. So how do you explain this concept of incomplete dominance? With a very suitable example, that is Snapdragon, or also you can call it as Antiherem species or 4 o'clock plant. So what does this incomplete dominance state? So when the two heterozygous alleles are together, one of the dominant allele fails to suppress the recessive allele completely. As a result, both the alleles together express intermediate character. Example, I told flower color in Snapdragon, or you can also call it as Antiherem species or dog flower or 4 o'clock plant or Mirabilis jalapa. Now let us see the schematic representation. So, the phenotype of F1 generation does not resemble either of the two parents, and the expression is in between the two. It is noticed in the inheritance of flower color in the dog flower or Snapdragon, which is a very good example for occurrence of incomplete dominance. So, how do you represent it schematically? It is a cross between two true breeding red flowered plant and true breeding white flowered plant. So, here you can see red color flowered plant is crossed with a white flowered plant. The F1 produce pink color here. The F1 produce pink color. According to the law of dominance, the color should have been red. But instead of red, there is an intermediate color. Pink is neither red nor white. So, as a result, when the F1 hybrids are self-crossed, that is, the F2 generation resulted in the ratio of 1:2:1, that is 1 red is to 2, pink is to 1 white. Here the phenotype ratio does not resemble the Mendelian monohybrid cross, but the genotype ratio is exactly similar to that of Mendelian monohybrid cross. So already we have studied that inheritance of one gene which is explained with Mendel's monohybrid cross, the phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1. But here you can see that in this particular schematic representation which explains the incomplete dominance, the phenotypic ratio as well as the genotypic ratio is 1:2:1. That is 1 red is to 2 pink is to 1 white. That is what is being told here. So let us see the schematic representation. Parental generation, homozygous red color flower plant is crossed with homozygous white flower. So the genotype is capital R, small r and smaller. So gametes capital R and smaller. So the F1 generation, when you cross it, instead of getting red, you get a pink color. How do you go for F2? F2 is obtained by crossing selfing. F2 is obtained by crossing F1 into F1. So here, we'll see in the next slide. So here, Punnett square. So when we use this Punnett square, it is easy for us to understand. So showing gametic combinations and the natural and the second filial generation. So if you take gametes, male gamete on one side and female gamete. So this is 1 F1 and this side it is 1 F1. So here when you cross capital R, you get red color, capital R, small r, that is pink color. Again cross this, you get pink color. and when you cross this, you get white flower. So here phenotypic is what? 1 red flower, 2 pink flower, 1 white flower. And genotypic ratio, 1 red flower, 2 pink flower and 1 white flower. So this is a very important concept from examination point of view, which usually appears in the question paper. So you need to study this schematic representation of incomplete dominance, as well the monohybrid cross and the dihybrid cross. Moving on to the next question, on which bacteria Griffith conducted transformation, that is Streptococcus pneumoniae. Then explain the mechanism of XO type of sex determination in insects. So in insects, you can see the parents female AA plus XX, male X0. So when you cross, so what are the gametes? You get A plus X. Here A plus X, A plus O. Here cross it with this, you get AA, XX, AA, X0. So this will appear as what? So here you can see this is female and this is male. So here in this type, the females are homogametic and have XX parents. The chromosomes produce only one type of egg carrying X chromosome, while the males are heterogametic and have only one X chromosome without the partner. Hence, they produce two types of sperms, where 50% of the gametes carry X chromosomes with autosomes AA plus X, and offspring other 50% of sperms carry autosomal but no allosomes. So XOX type of sex determination, which is the classical example, is grasshopper. Moving on to the next question, question number 14. Distinguish between homozygous and heterozygous plants. Homo means what? Similar. Hetero means what? Dissimilar. Plants having identical pairs of hybrid plants having alleles or allelic pair of genes. Hybrid plants having alleles expressing contrasting traits or dissimilar alleles is called heterozygous. Next, what is test cross? Write its significance. For example, like if they ask the test cross for monohybrid cross, it is 1:1. And if they ask the test cross for dihybrid cross, it is 1:1:1:1. So it's a kind of back cross where F1 plant is crossed with its recessive parent. So F1 into parental recessive is called test cross. What is back cross then? Back cross is equal to F1 into either of the parent. Either of the parent. It might be the dominant parent or the recessive parent. So it is a kind of back cross where F1 plant is crossed with its recessive parent. It helps in finding what? F1 plant is either homozygous or heterozygous. Next, write a note on color blindness. It is a sex-linked recessive disorder due to defect in either red or green cone of eye, resulting in failure to discriminate between red and green color. This defect is due to the mutation in certain genes present in the X chromosome. It occurs in about 8% of males and only about 0.4% of females. Next, what are Mendelian disorders? Give two examples. Already I explained in the previous slide, genetic disorders have been broadly classified into Mendelian disorders and chromosomal disorders. So what are Mendelian disorders and what classical examples you are going to give for Mendelian disorders? Gene disorders or Mendelian disorders are mainly determined by alteration or mutation in the single gene. These disorders are transmitted to the offspring on the same lines as we have studied in the principle of inheritance. Most common Mendelian disorders are what? Hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria and thalassemia. The examples are very, very important. Next, why has T.H. Morgan selected fruit fly for his genetical experiment? Already I had explained in the previous slide. Once again, they could be grown on simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks. A single mating could produce large number of progeny flies. There was a clear differentiation of the sexes, that is, sexes are separate. The male and the female flies are easily distinguishable. Now, question number 19. Write the karyotype of the following syndromes. Karyotype means the chromosomal. This can also be called as chromosomal complement. So Down syndrome, how do you write? The chromosome 45A plus XY, that is male. In case of female, 45A plus XX. This chromosomal complement is very, very important from examination point of view. This is very important. So when you are explaining the syndromes, particularly the chromosomal syndromes, you need to write the chromosomal complement which gives you the clarity about that syndrome. So Klinefelter syndrome, already I told, Down syndrome is common for both male and female, and Down syndrome is due to trisomy. Klinefelter syndrome is also due to trisomies. That is the reason we have 47, 47. So here 44A plus XXY 47. Turner syndrome is an allosomal disorder. Klinefelter syndrome is also an allosomal disorder, but whereas Down syndrome is an autosomal disorder, or it is also called as what? 21st trisomy. So with XO, that is 44A plus XO, that is 45. This is due to monosomy, that is 2n minus 1, and these two are due to trisomy, that is 2n plus 1. Next, what are multiple alleles? Give an example. Write the symptoms of Klinefelter syndrome. So here we asked the chromosomal complement, but it's a split question which is asked the symptoms of Klinefelter syndrome, which is seen only in males. So what are multiple alleles? Give an example. The expression of genes in more than 2 allelic forms of a character called multiple allelism. Example, the occurrence of ABO blood group is a classical example for multiple allelism. Now, what are the symptoms of Klinefelter syndrome, which occurs only in males? Individual has overall masculine development. Gynecomastia, that is feminine characters like absence of face. Gynecomastia means development of breast. Feminine characters like absence of facial hair and presence of enlarged breast. So enlarged breast is also called as what? Gynecomastia. Underdeveloped testes, absence of spermatogenesis and sterility. Now, 21st question. The shape of RBCs of sickle cell anemia patients change to elongated sickle-like structure. Give reason. Why? The mutant hemoglobin molecule undergo polymerization under low oxygen tension, causing the change in the shape of the RBC from biconcave disc to elongated sickle-like structure. Next, significance of test cross. Why test cross is done? Why it is so important? Already I told, test cross is a cross between F1 and parental recessive. It helps in knowing whether the organism is F1 is homozygous or heterozygous for a trait. It helps in knowing the genotype of an individual for a particular character. It helps in predicting the genotype of the F1 plant. Then what is male heterogamity? Explain sex determination in humans. Male heterogamity. Males produce 2 types of gametes. Sperm with respect to determination of sex is called male heterogamity. So how do you determine the sex determination in human? Human is what? XX and XY. So one of the X fuses with another X. It is female. One X with one Y, that is male. So the sex determining mechanism in case of human is called as what? XY type. Out of 23 pairs of chromosomes present, 22 pairs are exactly same in both male and female. That is, autosomes are same, 44. The only difference lies in XX in case of females and 44 plus XY in case of male. So the only difference lies here. This is common. Autosome is same. 22 pairs means what? 22 pairs means 44 chromosomes. A pair of X chromosomes are present in the female, whereas the presence of X and Y chromosome are determinant of the male character. During spermatogenesis among males, 2 types of gametes are produced. 50% of the total sperm produced carry the X chromosome, and the rest 50% has Y chromosome besides the autosome. So females, however, produce only 1 type of ovum with an X chromosome, and there is an equal probability of fertilization of the ovum with parents, the sperm carrying either X or Y chromosome. Then in case the ovum fertilizes with the sperm gametes carrying X chromosome, the zygote develops into a female, already I told, and the fertilization of ovum with Y chromosome offspring carrying sperm results in male offspring. How do you write it schematically? So male, how do you represent the chromosomal complement? 44 plus XY. Female, 44 plus XX. So gametes, how do you write? 22 pairs means what? 44. Here 22 plus X, 22 plus Y. Here when you split the gametes, 22X plus 22, here it is X, ova, 22X, 22X. So 44, here it is X. So here you can see female progeny, 44 plus XY means what? Male progeny. This is the schematic, and this is also very important from examination point of view. Hope you all have understood the important concepts and the questions that might be probably asked in the coming examination of this particular chapter, chapter 4 of your biology syllabus, called Principles of Inheritance and Variation. So the questions what I had revised in this particular session pertains to the questions that were previously asked in the previous question paper. Hope I am clear with this particular chapter and the concepts what I have explained in this particular chapter called Principles of Inheritance and Variation. Thank you.